Well, hello and welcome back for part 2 of chapter 27, which is on fluids in the body and a different way of conceiving of those fluids and of studying them and understanding them. And we've gotten through the general relationships, the importance of osmosis, the concepts of intracellular fluids, the cytosol and extracellular fluids, the interstitial and plasma components of fluid in the body, how they stay in balance because of this diffusion relationship of water across membranes. So it's an opportunity now that we haven't had in a while of taking a separate look at the electrolytes and the molecular components, the ions that are present in the body, and recognizing some of their special roles. And we'll start with sodium, major component of salt. We get it by diet. The only living thing, the only thing that we eat regularly that's not alive is salt. So sodium and chloride are going to be ready supplies of pure and balanced ionic increases when we eat salt. Sodium ion is the dominant cation in the extracellular fluid. And so we've talked about this so many times, the sodium-potassium pump, its importance in cell potentials and transmembrane potentials, its importance in muscle and nerve actions, that when we poke a sensitive membrane, what we often do is open a sodium gate. And sodium rushes across. Its high concentration outside the cell compared to its low concentration inside means that diffusion alone pushes it through that gate. But more important than that, the increased protein content of the cytoplasm produces an electrical attraction. Now, I can't give you an actual voltage, but just understand this. If there's much more negative charge inside than outside, the relative electrical force is to pull the sodium in. So that sort of moves in quickly. 90% of the extracellular fluid's osmotic pressure is due to the sodium content. And its main form, sodium chloride, is important, but because of carbon dioxide production, sodium bicarbonate. So what we see here, normal sodium concentration, if we increase sodium levels in the extracellular fluid, and homeostasis is disturbed because we're near that upper limit, it affects the hypothalamus and its receptors for osmotic pressure. This secretes ADH, which restricts water loss and stimulates thirst in connection with other things, and basically promotes additional water consumption. Because we have added sodium, a couple of shifts happen in the body. Look at this. It says osmolarity increased, water shifts out of the ICF, increasing the ECF. Now that increases the extracellular fluid volume and decreases the sodium concentration. Now you can also imagine if you're recruiting, you're recalling fluids out of the ICF and into the ECF, you're increasing the interstitial fluid and the plasma volume, so the pumping is improved. This decrease in sodium restores homeostasis. If we go the other way, we're losing sodium, so a low sodium level, we inhibit this, ADH secretion decreases as soon as the osmotic concentration decreases by 2%. So basically, we're not thirsty anymore. We don't drink. And water losses in the kidneys, as ADH is used up, goes away. Water loss in the kidneys decreases. This decrease in increased water loss decreases the extracellular fluid volume. That increases the sodium concentration and restores homeostasis. Hyponatremia is low sodium content. Overhydration is the main cause. So you want to dump some body fluids. And this happens when the ECF, sodium concentration, is below 106 milliequivalents per liter. Hypernatremia, high sodium concentration, is when the body water content declines. This is dehydration, and this is where sodium concentration is greater than 145 milliequivalents. Plasma volume affects, of course, this entire system. So sodium being the main osmotic regulator of extracellular fluid. That venous return, if the volume of the plasma goes up, stimulates the release of ANP and BNP, reduces thirst, increases the passing of water, bringing that volume back down. Salt and water loss, by the way, is something we can regulate at the kidneys. And we basically start putting more sodium into the urine, and the amount that's expelled goes up, and the extracellular fluid declines. So looking here, increasing ECF by fluid gain or by fluid and sodium gain. You remember we said pump the salt and the water follows? The same thing applies to retain the salt. If you retain the salt, it keeps water in the body, and blood volume goes up. You can see the organ effect here. So increasing blood volume and atrial distension. Now we have things like stretch receptors and pressure receptors that are monitoring this, and blood pressure starts to go up, atrial stretching starts to go up. Natriuretic peptides have these effects. We increase sodium loss in the urine, largely through a decrease in reabsorption. Increased water loss in the urine. So if you keep the sodium in the filtrate, it keeps the water there. Thirst decreases. And we also inhibit those things that cause retention. Basically, the increased volume of low concentration urine moves the blood volume down, which decreases the blood pressure. And this ECF volume adjusts to be in normal range. The other way, decreasing blood pressure and volume, we have seen many times. And the ECF effect is fluid loss or your fluid and sodium loss. If you go too close to that bottom boundary of sodium concentration, you're going to see a drop in blood pressure and blood volume. This is where you get the renin, resulting in the angiotensin path to aldosterone and ADH, all of which have an effect on increasing the retention of sodium, retention of water, decreased urinary water loss, so lower concentrations of urine that have very high concentrations of solutes, increased thirst and water intake. So we're increasing the volume of the plasma just by drinking and restoring the normal ECF volume. Potassium, 98%, is in the intracellular fluid. This is a metabolic complement, a cofactor, a requirement for the activity of certain enzymes. It's explained by the sodium-potassium pump, and it has a big role in cell potentials. When we fired a nerve or when we moved a muscle, potassium was moved. Cells expend ATP to concentrate this. This is an active transport mechanism, and its balance is basically digestive balance. How much do you take in versus what you lose, principally in the urine. So factors that promote low potassium, hypokalemia. Now potassium is K because it's kalium. Diuretics like Lasix cause potassium loss in the urine. Aldosteronism, which is basically a result of increased aldosterone on a regular basis, results in potassium loss by overstimulating the reciprocal effects of sodium retention and potassium loss. Now, hyperkalemia, basically a dangerous condition of increased potassium levels. Low blood pH promotes this because low blood pH interferes with calcium excretion by the kidneys. Kidney failure can result from hyperkalemia, and there are several drugs that promote diuresis by blocking sodium reabsorption. And when this happens, then potassium secretion also goes down. Calcium. Calcium is kind of, as I studied anatomy late in life and physiology, it kind of became my favorite ion. It did so many things I was not aware of. Now, we have a lot of calcium. We actually have more calcium in the body than sodium and potassium, but that's not an electrolyte effect. That's a skeletal effect from the fact we're vertebrates. We make hard bones. Most of our calcium, and you can see that a typical individual has up to 4.5 pounds of this. Over 99% is deposited as the crystal mineral hydroxyapatite in making our hard bone. That leaves very little left over as soluble calcium, 8 to 11 milligrams per deciliter, but it also provides a pretty handy relationship. If our calcium gets low, we can basically just chew up a little bone and dump the calcium into the circulating blood plasma. If it gets a little high, we can basically bump those osteoblasts and basically say, well, over here, just make a little more bone, mineralize the bone. So since we have a ready place to store it and a ready place to get it, calcium is kind of as a backup of over and above dietary supply. Now, calcium, as we remember, was involved in the release of the sarcomere. Remember that the troponin and tropomyosin locked that sarcomere so that the myosin couldn't bind with the actin site. It was calcium that unlocks that. So to contract a muscle, you need the right concentration of soluble calcium. At the synapse, you remember that the arriving action potential caused a calcium gate to open and flood calcium into that telodendrion on the presynaptic side of the synapse. Now, its effect has caused the neurotransmitter vesicles to fuse and dump their neurotransmitter. So if you want to move a nerve impulse, you need calcium. Very, very low levels, but also look at this, a very, very narrow range. So I think in other places it's recorded as 8.5 to 11. If it goes out of kilter more than 2.5 milligrams is its full range, then the body works to bring it back into range. Magnesium is something we haven't mentioned specifically, as I said earlier. It is important in bone. We do put magnesium in that crystal. We talk about hydroxyapatite as being a calcium carbonate and a phosphate salt, and it is, but it contains traces of others. One is magnesium, 29 grams of magnesium. About 60% of that is in our bone. But magnesium ion plus 2 is very important as a general cofactor. It's needed to phosphorylate glucose. Those initial steps of glycolysis where we were sticking phosphates onto the ends of glucose before we split it requires magnesium. Who knew? When we use ATP to contract muscle fibers, and you remember that 4-step process where the myosin would bind, power stroke, release and cock again. That four-step process. Every time we use an ATP, we need magnesium as a cofactor. Magnesium comes back in to the bloodstream pretty effectively in the proximal convoluted tubule of the kidney. So we reclaim almost all of it. But we do require some in our diet to balance that part that's lost in the urine. You know, 0.3 to 0.4 grams of potassium is ordinarily lost. Phosphate ion, gosh, we say phosphate so many times. Every time you hear ATP, every time you hear DNA, DNA is made of nucleotides, which is a sugar phosphate backbone with organic bases. It's required for bone mineralization, a part of hydroxyapatite. About 3/4 of a kilogram of phosphate is found in the skeleton. Urinary and fecal losses of about 0.8 to 1.2 grams. In the intracellular fluid, in the cytosol, phosphate is very busy. High energy compounds like ATP and GTP, the P there is phosphate. Phosphate there to activate enzymes so they can actually perform their metabolic conversion. Any energy requiring reaction uses phosphate. And the synthesis of nucleic acids uses phosphate. So it's basically in the plasma, reabsorbed from the tubular fluid along the proximal convoluted tubule. Again, you know, that PCT, we talked about filtration and then an immediate reclamation of those things that are essential to us. So we've already seen several of these. Magnesium, phosphate, sodium, effectively reabsorbed early in that urine-making process. Chloride ion is not usual for us to talk about the negative ions. It's certainly the most abundant because we're eating it all the time. There's so many things that depend on sodium, but every time we eat an ion of sodium, we get an ion of chloride too. It's the most important balancer of co-transport, that chloride shift. And when you think about chloride shift, you should think bicarbonate. That's the major negative ion that we're making on a constant basis, which just basically is contributing to an excretion or an elimination cycle. We got to get that CO2 to the lungs to breathe out. We got to get that bicarbonate and CO2 to the kidneys so we can get rid of the extra hydrogen ion. And so chloride ion is involved in the cardiovascular stage of that. You got to pick it up and carry it somewhere else. Well, we're going to put the bicarbonate over here, but to balance the charge, we're going to bring a chloride in. That's called the chloride shift. When it's time to bring it out of the cell and back to the plasma, we're going to shift back. Usually chloride concentrations are low. Since both proteins and bicarbonate are abundant in the cytosol and are negatively charged, we basically don't incorporate much chloride into the cytosol. We do get chloride as salt in our diet, and it's reabsorbed along the renal tubules for retention. But chloride is another ion that contributes. Being outside the cell contributes to the osmotic pressure in cells. Hydrogen ion, gosh, in terms of ion, it's kind of notable in several regards. It is very reactive. Because although we talk about it with the name of an element, hydrogen, H plus is just a proton, isn't it? Just a bare proton. So it is this massive subatomic particle, but it's also highly charged. It's got a +1 charge. It doesn't share that charge with any other subatomic particles, just a single proton. Very, very reactive. We notice its presence causes chemical reactions. And as a result, we track it with the pH, or power of hydrogen ion, in the body fluids. It's from 0 to 14 in water, and it's intimately associated with the production of CO2 and water intimately associated with acid production. We do have a number of acid systems in our body. If a strong acid or a strong base, that means they totally dissociate in water. So a strong acid would exist totally as, for instance, hydrogen ion and fluoride ion, or hydrogen ion and chloride ion would be completely dissociated. That's what makes it a strong acid. A weaker acid, like acetic acid, H2CO3, you'd get the H plus and the bicarbonate in equilibrium, but there'd be still a lot of H2CO3 in that solution, so the pH would be somewhat higher. They would be weak acids or bases, and these are the ones we use in buffer solutions. Buffer solutions can absorb hydrogen ion fluctuations and maintain a consistent pH. Fixed acids. You produce, if you have a sulfur or a phosphorus source in our body, you can produce things like amino acids, nucleic acids, phospholipids. These are regularly required fixed acids. They're basically, if there's an excess, they stay in solution until the kidneys take them out. Organic acids from our aerobic metabolism. They do not build up in our bodies. Lactic acid from anaerobic metabolism does build up and is harder to remove. That would take, if you have an anaerobic exercise, you're going to get sore. Overstraining your muscles, you're going to get sore. It's going to take a matter of days to clear that out for that soreness to disappear. But normally, this organic acids from aerobic metabolism, CO2, we are busy transforming them to the bicarbonate carrier, moving them to the lungs and breathing them out, secreting the excess into the urine and removing it by excretion. Volatile acids from CO2 is a special category, and its exit is part of our pH metabolism. We gain hydrogen ion across the digestive tract and through cellular metabolic activities. And we already talked at length about the production of CO2, but I want to go back to this first one, the digestive tract. We actually like sour foods. And there are so many foods of high value that are sour. We call it tart. Sometimes we call it outright sour. So the pH of something like a tomato or a grape, anything that has a tart flavor, is around pH 4, pH 5. Citric acid in lemons, in limes, in oranges, in grapefruit. Although those tastes are slightly different, the pH is a pH 4 to pH 3. Soda pop, carbonic acid in soda pop, pH 2. And we take these foods in great quantities, which tells you that our pH regulating mechanism is very adept. We eliminate at the kidneys and at the lungs based on that CO2 system that we talked about. If excess acid builds up, we do get pain, and it's basically a very simple thing. The acid irritates a pain receptor and it sends a message. But it is part of our normal metabolic activity and elimination. We do manage this pH issue. There's so many things, producing hydrogen ion and introducing hydrogen ion. And we have a number of chemical components that can act as buffers. Now, what a buffer does is it sits at a certain pH, and if hydrogen ion is added, it can absorb that so that the hydrogen ion, the general hydrogen ion concentration does not go up. But this is the way a buffer works. So we see in the intracellular fluid, we buffer mainly with phosphates and with proteins. Inside the cell, the phosphate buffer system of PO4 to HPO4 is something that can absorb and manage excess hydrogen ion. Here in the protein buffer system, we have hemoglobin, which can absorb hydrogen ion in red blood cells and be that hydrogen transport mechanism back to either the lungs or the kidneys. amino acid buffers. Amino acids can absorb hydrogen ions. Proteins in general are negatively charged. So what does that mean if a protein is out there and each molecule has a -4 charge at physiological pH? That means it can easily bind with hydrogen and carry it somewhere else. Amino acids and protein, plasma proteins out here in the extracellular fluid can also bind to hydrogen ion. Now, the plasma proteins have been mentioned before, like albumins, globulins, and fibrin, mainly albumins out here, and bind hydrogen ion. And in the extracellular fluid, we do have a significant, we called it the alkaline tide. We used the chloride cotransport to set up this carbonic acid bicarbonate buffer system that transports this form of carbon and its relationship to hydrogen ion around the body. Hemoglobin buffer system, that's the CO2 in the red blood cell. No transport is required. As carbonic acid basically breaks apart to bicarbonate, hydrogen is released. Bicarbonate, the alkaline tide, goes out into the plasma for transport. And you saw that hemoglobin did bind hydrogen ion inside the cell. So there is some buffering there, but again, this is temporary. Pick it up, carry it over here, shove it out. Come back around and pick up another load. So we see this here, CO2, and its complicated hydrogen relationships. CO2 producing carbon dioxide binding to hemoglobin. Now, this carbon dioxide and this carbon dioxide accounts for only about 25% of the CO2. With water in here, you make carbonic acid, and here's the hydrogen ion. A whole bunch of it binds to hemoglobin, and the bicarbonate gets shoved out, chloride brought in for the transport. At the other end, you see a reversal of this. The co-transport, chloride out, bicarbonate in, combining to carbonic acid, which remanufactures water. Now, over here, you remember we used water? So in terms of our overall water balance, we're giving that water back here, and it comes out balanced, yielding the CO2 so that we can shove that CO2 out. Hemoglobin gives up its CO2, and the CO2 from the plasma joins this parade of CO2 molecules out to gaseous form and expiration to the outside world. Acidosis results if you have hydrogen ion out of control. So how do we control that in the body if the hydrogen ion is built up to an intolerable level? Well, we can respond normally in two very specific ways. We can breathe harder and expel more CO2. That actually consumes hydrogen ion, because in converting from here over to CO2, you're actually making water. Water doesn't contribute to the pH effect. The bicarbonate reserve is one that can absorb hydrogen ion and form bicarbonate reserve. Sodium bicarbonate basically takes care of this excess bicarbonate. The kidneys can respond by increased secretion of hydrogen ion, removing the CO2 and reabsorbing the bicarbonate to help replenish this bicarbonate reserve. Here's another use for our sodium ion. Alkalosis, if your pH goes up, we basically can neutralize that base. And by the way, whereas we can tolerate some fluctuation of blood pH and local pH conditions, when the pH goes down from 7.4, the pH goes up, we have a very narrow window of response. So over here in the lungs, we decrease the respiratory rate and basically enhance the CO2 level in the body, driving the equilibrium in this direction. The hydrogen ion will then neutralize the excess base. The presence of in the kidneys, we conserve hydrogen ion and we secrete bicarbonate, retaining the acid in the body and neutralizing the base again. I'm sorry, these are all described here. Respiratory acidosis, when the respiratory system isn't clearing the CO2, the pH of the plasma goes low, and the main cause, hypoventilation, breathing at too slow a rate. Metabolic acidosis is somewhat different. 3 causes. If we have large production of fixed and organic acids, the hydrogen ion just builds up. This is where you go anaerobic. Lactic acidosis occurs. Lactic acid is hard to metabolize. Ketoacidosis, a different conversion to produce a form called ketone bodies is seen. The ketone body concentration goes up in the blood. This is a problem during starvation or during diabetic attacks when there's a specific problem in digestion. The second cause, hydrogen ion secretion and excretion in the urine is impaired by a loss of kidney function. And the third is a severe bicarbonate loss and a loss of the buffering capacity. So this is the conclusion of chapter 27. We're doing very well with respect to our progress through the lecture material.